afternoon, ladies and gentlemen. Thank you very much for the time. As representative author, I would like to address our research entitled Synthesis of Piperine and My Derivatives and Evaluation of their Anti-Inflammatory Activity. My name is Dina and let's get started. First of all, before going to the bottom line, let me explain about the outline of this presentation. This will be divided into two major parts, introduction and experimental, including method results and discussion. In the introduction, we will discuss about inflammation-linked disease, the side effect of existing anti-inflammatory drugs that lead us searching for a new potential anti-inflammatory agent. In the experimental session, we will discuss about the isolation of piperine, the synthesis of piperine derivatives, as well as their evaluation of anti-inflammatory activity. Inflammation is body response to the cellular damage that can cause from external factors such as hurt, burn, or chemical irritation. The damaged tissue will activate the heart rate pathways such as angiogenic and immune system that embed into tissue to facilitate healing and homeostasis. If pro-inflammatory and anti-inflammatory factor in the system is balanced, it will undergo to the acute inflammation. Acute inflammation is a good sign because it will eradicate or speed up the healing process. In contrast, if inflammation turns up too high and the immune system continues to pump out the pro-inflammatory factor, including the nitric oxide, it will go to the chronic inflammation. Chronic inflammation has recognized as one of the significant leading death cause worldwide. It was reported in Nature Medicine in 2019 that more than 50% of deaths were attributed to inflammation-linked disease such as heart and kidney disease, stroke, and cancer. There were several medications that available such as corticosteroid type and non-steroidal anti-inflammatory drugs that is most common. However, these drugs will give negative effect to the patient as it mentioned in the slide. Therefore, more research are necessary in the field of discovery and development of anti-inflammatory with fewer side effects. In this past decade, there is growing interest in the using such a natural drug product as the emerging as a potent drug with fewer side effects. And one of the natural products is piperine. Piperine is an alkaloid that consists of three subunits, an amide linkage with piperidine moiety, butadiene chain, and 1,3-benzodiazole group. Piperine predominantly found in the fruits of Pipernicum or Piperlongum species of Piperaceae family with constituent around 2 to 7%. It was reported that piperine shows a wide range of biological activity such as antimicrobial, anti-inflammatory, anti-cancer, and antioxidant. More research are revealed about the mechanism of action of piperine as anti-inflammatory. It was reported by Ying and co-worker on 2013 that piperine suppressed the pro-inflammatory factors such as cyclooxygenase 2, tumor nuclear factor alpha, and inducible nitric oxide synthase. Moreover, Umar and co-worker on 2013 also reported that piperine inhibits the production of interleukin-1 beta and interleukin-6 that involved in the inflammation. Even though there is a lot of mechanism of action that revealed about the piperine as anti-inflammatory, as the base of our knowledge, there is still no study regarding the piperine derivatives and evaluation of their anti-inflammatory activity. Therefore, this work we will dealing with the chemical modification of piperine to an dimer and amide series and evaluate their activity as anti-inflammatory. So let's get started with the first one, isolation of piperine. The isolation of piperine is started with the extraction of fruits of Piperlongum L with methanol and then partitioned using ethyl acetate. The ethyl acetate extract then purified by column chromatography with solvent system hexane ethyl acetate 723 to yield 2.1% of piperine. The next step is synthesis of pipric acid. The pipric acid prepared by alkaline hydrolysis of piperine. The piperine was refluxed overnight at 20% of methanolic potassium hydroxide, and then the result suspended in the water and acidified with hydrochloric acid to reach pH 1. After that, doing extraction using ethyl acetate and evaporate to get the pipric acid. The next step is synthesis of amide series derivative. The conversion of an acid to an amide usually will involve oxalyl chloride or thionyl chloride. However, this method is dangerous because it will accompanied by the chemical hazards such as hydrochloric acid and also the carbon monoxide. Therefore, this research we using dicyclohexyl carbodiimide along with dimethylaminopyridine as a catalyst. First, the acid reacted with DMAP and DCC, and then the addition of amine allowed the formation of the imide. All of the synthesis compounds showed relatively low to moderate percentage yield ranging from 28.2 to 58.1 percentage yield. Compound number 6 showed lower percentage yield compared to compound number 3 and compound number 4. It's because of the amine of compound number 6 containing of aromatic ring system, which can decrease the nucleophilicity of the amine. And then compound number 5 had the least percentage yield compared to the others because of its steric hindrance from the amine. The next synthesis is synthesis of piperine dimer. The piperine dimer was prepared by Diels-Alder reaction of piperine by heating the piperine to 160 degrees Celsius for 6 hours in the presence of silica to yield 42.5%. The last step is bioactivity assay. Before doing the anti-inflammatory activity of all the compound determine its toxicity towards cancer, toward the cell lines. First, the cell was seated in 96 well plate and incubated for 24 hours at 37 degrees Celsius and 5% of carbon dioxide, and then addition of various solution or sample followed by 24 hours of incubation. And then addition of MCT followed by 4 hours of incubation, and then the last is addition of DMSO for formazan solubilization and measure the absorbance at 570 nanometers. The compound categorized as toxic if the cell viability is less than 80% at the dose of 50 micromolar. The anti-inflammatory activity was determined by monitoring the nitric oxide inhibition of LPS-activated macrophage J774A1 cells. First, the cells were seeded and incubated for 24 hours at 37 degrees Celsius and 5% of carbon dioxide. And the next step is treated with sample and doing incubation for 2 hours. Next, activate by LPS and incubate for the next 20 hours at the same condition. The concentration of nitric oxide were determined by Gris reagent following this reaction, and the absorbance then measured at 540 nanometers. The result reported in the IC50 value. If IC50 value lower, it means it's better. This is the result of the cytotoxicity evaluation and anti-inflammatory activity evaluation. All of the compounds show no toxic to the cells except for compound number 7. In consequence, compound number 7 did not do for further anti-inflammatory activity. Piperine compound number 1 is a parent compound, show nitric oxide inhibitory effect relatively the same with indomethacin as the positive control with IC50 value of 44.4 micromolar. And then The functional group from piperine, such as an amide linkage, glutathione change, and methylene deoxy, was expected to give some interaction to an enzyme that involved in the nitric oxide synthesis, so it gives some activity. And then compound number 3 show the most potent if we compare to all the compounds. Compound number 3 and number 1 have the same, but the difference is just the ring size. The smaller ring size of compound number 3 probably will allow the compound to fit with the enzyme. The next compound that show good activity is compound number 5, followed by compound number 6 with IC50 value of 26.7 and 27.2 micromolar respectively. Compound number 4 showed moderate activity towards T774A1 with IC50 value of 38.9 micromolar. Compound number 5 has two butane change if we compare to compound number 4 that only have one butane change. The more aliphatic change will increase the lipophilicity of the compound, so it will allow the compounds to penetrate easily to the membrane cell. In case for compound number 6, it containing of aromatic ring system, but that contribute in the pi-pi stacking interaction between the compound to the enzyme. And the last is compound number 2 that show no activity towards no activity for anti-inflammatory. So from this finding, we can conclude that the amide linkage of piperine derivatives probably play a key role in the anti-inflammatory activity. So in the conclusion, we got piperine was isolated from the fruits of Piperlongum L, and then piperine amide derivatives 3 to 6 were prepared to TCC coupling method. Meanwhile, compound number 7 was prepared by silica-mediated Diels-Alder reaction of piperine. The piperine and its derivative show better activity than indomethacin as the positive control with IC50 values ranging from 19.5 to 44.4 micromolar. Except for compound 7 that is toxic to the cell, so we did not do for anti-inflammatory activity. The last but not least, I would like to thanks to my advisor, all of the committee, and all of the audience. Thank you very much.
Thank you very much for the good presentation. Are there any questions from the audience? Yeah, I have one question I can catch you up. Okay. Why the compound number 3 is the most effective for anti-inflammatory?
Compound number 3.
Yes, why compound number 3 was the most effective for anti-inflammatory?
Okay, if we see compound number 3, it have like the ring size is, if we compare compound number 3 and compound number 1, the compound number 3 have the ring size smaller than compound number 1. The smaller size probably will increase the possibility to fit with the active site of the enzyme, Kajan.
You mean that the small ring, this ring, okay?
Yes.
This is the bi-active site of the enzyme?
Probably, because we do not do for further mechanism, so we just guess from the structure activity relationship.
Okay, thank you. And any question? Yes, they're very interesting and very useful research.
Thank you, Kajan.
If you know any question from the audience, thank you very much, Dina. Thank you very much.